IB students. All right, so today we're going to finish 9.4 and hence we finished topic 4. Topic 9, sorry. So after topic 9, notice that we're going to be moving on to topic 10, which is genetics. So looking forward to that. That should move along rather quickly. And then my favorite, animal physiology, starting with the oh so interesting immunology or antibody production. So really looking forward to that. Okay, so today we are going to look at seed structure. This is very simple. The skill is to be able to draw the internal structure of seeds. So pretty much what you're going to be drawing is something that looks like this picture here. So let's take a look. Get my pen here. All right, so we know that when fertilization occurs, the ovule, which is the egg, will develop into a seed, which is inside a fruit. And then that seed is going to get dispersed, and then that seed needs to germinate. That means it needs to give rise to a new plant. So let's look at the internal structure of a seed. So let's pretend we took this seed and we did a cross section. So we opened it and we are looking at the inside of the seed. So first we start with the testa, which is the outer seed coat that protects the embryonic plant. Then we have the micropyle, which is a tiny little pore here. And this is on the outer covering of the seed, and it allows for passage of water, because when the seed germinates or receives water, the water goes in through that tiny little hole. Then we have the cotyledon. The cotyledon is what contains the food storage for the seed, and this is where the embryonic leaves are going to arise. So when we think of the cotyledon, we think of seed leaves. And you've heard of the term monocot, which means one seed leaf. And you've heard of the term dicot, which means two seed leaves. So pretty much that tiny little seed that's going to form here, and I'll draw a picture in just a minute showing you what it would look like, is the cotyledon. That's it. And then the plumule, which is the embryonic shoot, and this is also called the epicotyl. And the embryonic shoot, or the epicotyl, or the plumule, this is this region right here, and this is going to give rise to the shoot of the plant. And then we have the radicle right here, and this is what's going to give rise to the root of the plant. Okay, so if you were asked to draw this by hand, and again, I'm a terrible drawer, you would draw something like this. You would draw the outer covering, which is the testa, and I'm sure your picture would look a lot better than mine. But then you would draw the interior here. And usually what I do, if you know, I'm not good at drawing, I just do something like this, okay? And then this in here would be the cotyledon. This up here would be the epicotyl. This down here would be the radicle and epicotyl. And I would draw a tiny little pore right there to show the micropyle, and that's it. Now, interestingly enough, if you were to look at a cross section between a seed, that, let's say I take the seed and I open it like a book, right? And we would be doing this in class, in a lab. If I were to open it like a book, it would look something like this. Okay. Here's the other one. All right, and if I see two cotyledons here, then I would assume that that is a dicot plant. Now, if water got in there through the micropyle, then you would see a swollen cotyledon. If you were dissecting a dry seed and you open it up like this, um, sometimes you see the cotyledon completely dehydrated and shrunk. So if you open up a seed with no water, then you're going to see those shrunk cotyledons, if it's a dicot plant, of course. So if we're doing this in the lab, we can add water to a seed. We can leave it in water and we can see what the cotyledons look like. And then in contrast, we can look at a seed. It'll be very hard to open if it's dry, but not impossible. We could look at the cotyledons if no water has been able to go in. And then there's a variation of seeds that you can look at, right? You can look at the difference between monocot seeds and dicot seeds and see the 2 cotyledons versus the 1 cotyledon. Okay, the next part of this section is to design an experiment to test a hypothesis about factors affecting germination. So you might get a question in the form of an experiment that's already been designed and you have to analyze it, or you have to design an experiment on your own via a short answer question. So we know that germination is the process by which a seed emerges from a period of dormancy and it begins to sprout. But for germination to occur, there is certain requirements right here. So these requirements, if we move down here, we'll see that these are essential requirements or essential conditions. Other seeds have specialized conditions, and we'll look at those as well. But let's look at some of the essential conditions. So for a seed to germinate, it needs oxygen for aerobic respiration. It needs water to metabolically activate the seed and trigger the synthesis of gibberellins, which is another plant hormone, but we didn't talk about it. It needs to be at an optimal temperature in order for it to sprout for optimal function of enzymes. It needs to have an optimal pH in order to sprout, again, for the optimal function of enzymes. And additionally, certain plant species require specialized conditions. So some seeds will only sprout if they're exposed to heat, for example, a bushfire. Some plants will only sprout if they're frozen or they're put in intense cold conditions. So for example, you know that tulips need to undergo really cold weather in order to bloom the next spring, which is something that did not happen to my tulips in my garden last spring because the winter was really mild. Digestion. Some seeds require prior animal digestion to erode the seed coat before the seed will sprout. So the digestion and the hydrochloric acid inside the ruminant is going to help remove that seed coat. Some seeds required washing to remove some inhibitors that will inhibit the germination. And some seeds require scarification, which are seeds that are more likely to germinate if the seed coat is weakened from physical damage. But these are a certain plant species. So this would make for an excellent IA. This would make for an excellent experiment. But you have to do some research because not all seeds, not all plant species will germinate based on these conditions, right? You can't just scar any seed and expect scarification to germinate every single plant species out there, right? So careful research needs to be done before that. So any of these experiments can be developed using any of these factors as an independent variable, and then you can measure your dependent variable, the rate of seed growth over a set period of time, and make beautiful line graphs. Okay, so that is it for topic 9.4.